You've probably taken Tylenol, maybe this week. It's one of the most studied drugs in human history. We still don't fully understand how it works. If that sounds impossible, you've fallen for the same myth that makes health influencers think they can outsmart your doctor by reading a study. There's this unspoken belief about how medicine works. Scientists in labs do basic science, they discover mechanisms. That knowledge gets taught to doctors, and doctors then make recommendations based on that learning. It's clean, logical, makes sense, and for most of medical history, advances have happened exactly the opposite way. The story of Tylenol, how it went from an industrial waste to your medicine cabinet, is one of the most fascinating examples of how medicine actually advances. And it perfectly illustrates why the influencer's favorite move, cherry-picking basic science to contradict clinical practice, is not just wrong, but fundamentally backwards. Let me show you. The story of Tylenol doesn't start in a medical lab. It starts in the 1850s with industrial smog and streetlights. The plants that produced coal gas for lighting also produced a toxic black sludge as a waste product, coal tar. The companies wanted to find a way to sell it instead of dealing with the toxic waste, and finally got a lead when chemists learned to make dyes from some of the organic molecules. A massive German synthetic dye industry, led by firms like Bayer, was born from learning how to turn this black sludge into vibrant colors. That success created a hunger for new ways to sell these organic waste molecules, and the companies looked to the emerging medical industry to see if they could find a use in suffering patients who will often pay anything, no matter how slim the evidence of benefit. And then, in 1868, a physician named Karl Wunderlich published his work standardizing the fever curve. He had taken over 1 million temperature readings, and for the first time, fever wasn't just a subjective feeling, it was a number on a chart. And that made fever itself a therapeutic target. Just like influencers of today, without controlled trials, medical practice of the time decided that this metric, because we could measure it and because it correlated with illness, must be controlled and minimized, and that doing so would naturally reduce the impact of the illness. And so by the 1880s, you have a dye industry with a massive surplus of cheap chemicals from coal tar and a medical community desperate for a magic bullet to bring that fever curve down. The first breakthrough in 1886 was a complete accident. Two doctors, Kahn and Hepp, were trying to treat a patient for intestinal worms with naphthalene, another coal tar byproduct and incidentally the same chemical that's used in mothballs. A local pharmacist made a dispensing error and gave them a bottle of a different coal tar chemical by mistake instead, acetanilide. The acetanilide had no effect on the worms, but the patient's fever, which they were tracking on their new Wunderlich chart, suddenly plummeted. They had accidentally discovered a new, potent anti-fever medication, predating aspirin by almost 10 years. The dye company Cal & Co. immediately marketed it as antifebrin, and it was a hit. There was just one problem. It was incredibly toxic. It caused a serious blood disorder called methemoglobinemia, which stops your blood from carrying oxygen, turning your lips and skin blue from hypoxia. This set off a race to find a safer antifebrin. In 1887, chemists at Bayer patented a new related molecule called phenacetin. They sent it to Germany's top doctor pharmacologist, a famous rock star medical influencer of his day, Joseph von Mering, who declared it effective and crucially less toxic since it caused almost no methemoglobinemia unless taken in high doses. But von Mering was systematic. He wanted to test the whole class. He had already tested the parent, acetanilide, and the new derivative, phenacetin. The next logical test was another related molecule, acetaminophen. This chemical wasn't new. It had been synthesized back in 1878 by an American chemist, Harman Morse, as an academic exercise. It was old, and most importantly, it was unpatented. Bayer had zero interest in it and refused to manufacture a purified sample. So von Mering had his university lab prepare a batch to test. This was the fatal flaw. Our best guess about what happened is that his lab synthesis was impure and contaminated with a precursor chemical, p-aminophenol, and that was a potent toxin causing the same methemoglobinemia as acetanilide. And I'm emphasizing the p-aminophenol here because, well, it's going to be funny later. When von Mering gave this contaminated sample to patients, he saw exactly what he expected. It reduced fever, but it also made them turn blue. In 1893, he published his conclusion. Acetaminophen was just as toxic as acetanilide, and phenacetin was the superior, safer drug. Because of his enormous authority, that expert opinion was accepted as fact. Acetaminophen was declared toxic and shelved for the next 50 years. So, for half a century, the medical world marched down the wrong path, all because of a single influencer's technical error. The story picks up again in the 1940s, not because of basic science, but because of a new public health crisis. The market had split. Ethical phenacetin was prescribed by doctors, while cheap, off-patent acetanilide was used in over-the-counter headache powders. A large population of headache powder addicts developed, consuming huge amounts and suffering from chronic, severe methemoglobinemia. This clinical crisis is what founded the rediscovery of Tylenol. Two toxicologists at the Goldwater Memorial Hospital, Bernard Brodie and Julius Axelrod, were tasked with a simple question: why was acetanilide so toxic? To answer it, they had to invent new methods to measure the drug and its metabolites in blood and urine. And when they ran their experiment, they solved the case. They found, along with Greenberg and Lester, that when people took acetanilide, their bodies converted it into two metabolites. One metabolite, aniline, correlated perfectly with the toxic methemoglobinemia. The other metabolite, acetaminophen, correlated perfectly with the analgesia, or the pain relief. They then found the same thing with the safer phenacetin. And they had it. They had just proved that acetanilide and phenacetin were actually precursors, not drugs themselves. Aniline was the toxic byproduct, and acetaminophen was the safe, active medication all along, which Brodie proved by administering pure, uncontaminated acetaminophen to subjects and demonstrating no methemoglobinemia. Von Mering's 50-year-old influencer error, born from a contaminated sample with no attempt at replication, was finally exposed. Now, you might imagine that this scientific proof would overturn the word of an 1890s influencer who was basically doing chemistry on a glorified kitchen table, right? Everyone would switch to start using acetaminophen instead. The world would change. It did not. Phenacetin was already entrenched in the market as the safe medication for adults. The medical world ignored Brodie's proof for decades. This is both an indictment of physicians at the time for either failing to know or giving in to what patients were asking for, as well as an indictment of the process of medical decision-making via popularity, marketing, and influence. The final scientific chapter was again initiated not by basic science, but by clinical pathology. In 1953, Swiss physicians noticed an epidemic of fatal kidney-killing disease. They traced this renal papillary necrosis directly to its source: the chronic, long-term, heavy abuse of the safe phenacetin-based headache powders. It was in this environment in 1955 that Tylenol was finally marketed. But it wasn't as a safer phenacetin. The pharmaceutical manufacturer didn't believe they could compete with phenacetin because people are slow to change when they don't see an immediate harm. In a pure marketing move, McNeil Laboratories sold Tylenol Elixir for Children as a prescription-only alternative to aspirin, capitalizing on the different, visible, immediate problem of aspirin-induced Reye's syndrome and intestinal bleeding in kids. Phenacetin was entrenched in the market based on the expert opinion of von Mering, and because the harms of phenacetin took decades to develop. No one in the public thought there was any need for a safer medication, despite the science. It wasn't until 1983, 34 years after Brodie and Axelrod's paper, that the FDA finally banned phenacetin because von Mering, a popular influencer, had gotten it wrong nearly 100 years prior. So let's recap. Tylenol's rise was driven by industrial waste, a pharmacy error, an influencer's mistake, a toxicology study, a separate kidney failure epidemic, and a clever marketing campaign against a third drug. Then, finally, acetaminophen became a clinical and commercial success for decades, and for almost that entire time, nobody had any clue how it actually worked. The basic science was just beginning. So this drug was a mainstay. Now basic scientists get to play catch-up to provide clever rationalizations after the fact. The first major attempt to explain it came in the 1970s. In 1971, Sir John Vane made a Nobel Prize-winning discovery. He figured out that NSAIDs, like aspirin, work by inhibiting enzymes called cyclooxygenase, or COX, which produce inflammatory molecules called prostaglandins. This was a beautiful, unifying theory for an entire class of drugs. Logically, everyone assumed that Tylenol must work the same way. But it didn't fit. Yes, it was a potent analgesic, pain control, and an antipyretic, fever reducer, but it had almost no peripheral anti-inflammatory activity, and most importantly, it didn't cause the classic NSAID side effects. It didn't tear up your stomach lining or stop your blood from clotting. It was clearly different. The first big clue came in 1972 from Vane's own lab. In a paper with Robert Flower, they showed that Tylenol was a potent inhibitor of prostaglandin synthesis in brain tissue, but a very weak inhibitor in peripheral tissue, like the spleen. This was a landmark finding. It established that Tylenol's primary site of action was the central nervous system, CNS. But why? Why would it inhibit the COX enzyme in the brain, but not the same enzyme in a knee or a stomach? For 30 years, that was the puzzle. And then in 2002, researchers at Brigham Young University announced a discovery that seemed to solve everything. This is Exhibit A in the influencer fallacy. They found a previously unknown splice variant of the COX-1 gene in the cerebral cortex of dogs. They named it COX-3. It was the perfect suspect. It was found in the brain, and when they tested it, it was exceptionally sensitive to inhibition by Tylenol. The mystery was solved. Tylenol was a selective COX-3 inhibitor. You can imagine the headlines. Influencers in 2003 would have been all over this. Your doctor doesn't know this, but Tylenol works on a secret enzyme called COX-3. The latest science, a preclinical model, proves it. The excitement was, however, very short-lived. As other labs around the world tried to replicate the finding in humans, a critical problem emerged. They couldn't find it. The reason, it turned out, was a tiny but catastrophic difference in the genetic code between dogs and humans. In the dog gene, the piece of code that makes the new enzyme is 93 base pairs long. Since the genetic code is read in threes, this inserts 31 new amino acids and produces a modified but still functional enzyme. In the human gene, the same piece of code is 94 base pairs long. One extra base pair. That single extra letter causes a frame shift mutation. It's like a typo that turns the sentence, "the quick brown fox," into "nifx." The rest of the genetic message is complete garbled nonsense, and the result is that humans produce a truncated, non-functional protein that has no enzyme activity after all. So the definitive conclusion is a functional COX-3 enzyme, as defined in those dog studies, does not exist in humans, and thus cannot explain the effects of Tylenol. This wasn't just a simple mistake, it was a conceptual failure. It was a brutal, perfect example of the dangers of interspecies extrapolation, the exact fatal flaw at the heart of the influencer's basic science myth. So after COX-3 crashed and burned, researchers got smarter. They stopped looking for one magic enzyme and started mapping a cascade. And here's where we are today. We have two rock-solid findings from human studies. First, the POX model. That 1972 finding that Tylenol works in the brain but not the periphery was correct. The why is actually quite elegant. The COX enzyme has two active sites, a cyclooxygenase, COX site, and a peroxidase, or POX site. Traditional NSAIDs like aspirin or ibuprofen block the COX site. Tylenol doesn't. Instead, it acts at the POX site, and that can only happen if there's no peroxides around. The brain is a low peroxide environment, the rest of the body is not, and this explains Tylenol's unique profile. It works on COX in the brain, reducing fever, but has no anti-inflammatory actions in the rest of the body. Second, the serotonergic pathway. We have definitive proof from a double-blind crossover clinical trial in humans. In the study, when people were given Tylenol, it worked, but when they were given Tylenol plus a serotonin blocker, like granasetron or tropicitron, the analgesic effect was completely abolished. And this is profound. It proves that no matter what Tylenol is doing upstream, the final, indispensable downstream step for Tylenol pain relief in humans is the activation of this serotonin pathway. So those are our major human goalposts. The mechanism must be in the CNS, and it must end at the serotonin pathway. We also know a little about what happens to acetaminophen in between. As it turns out, for pain, acetaminophen is likely not doing the work itself. Just like phenacetin and acetanilide get broken down into acetaminophen in order to work, acetaminophen itself is converted to p-aminophenol. And you should recognize that name. This is the exact same chemical that contaminated von Mehring's batch back in 1893. The same molecule that's highly toxic in the blood turned patients blue and caused him to declare acetaminophen a toxic failure. The supreme irony of our current leading hypothesis is that this very same chemical, toxic in the bloodstream, is now believed to be the necessary precursor for pain relief from Tylenol in the brain. Somewhere, either in the liver or in the brain, we're not sure which, acetaminophen is converted into p-aminophenol, which in the neurons of the brain is then converted into the new active molecule AM404. And from there, we have data that in mice, AM404 hijacks the endocannabinoid system and increases activity at the CB1 receptor, which then sends signals down the spinal cord to activate serotonin release to block pain. And in mice, this is really compelling because if you breed mice that genetically lack CB1 receptors, Tylenol doesn't work. Case closed, right? Except we've been here before. This is the COX-3 trap all over again. We are once again looking at a compelling finding in a different species, a knockout mouse, and assuming that its biology is our own. An influencer today will tell you with absolute certainty Tylenol works by activating your CB1 endocannabinoid receptors, and they are making the exact same unproven leap of faith that proved so wrong in 2002. A mouse's endocannabinoid system is not a human's. A mouse's pain pathways are not a human's. It's an interesting hypothesis, but it is not a human fact. And so after 130 years, our current model of Tylenol is a Rube Goldberg machine, one where we know the input, we know the output, but the middle is still anyone's guess. We know from human data that Tylenol goes in, it works via the POX mechanism for fever, and then for pain, it's converted via p-aminophenol to AM404, and its final effect requires the serotonin pathway. We hypothesize from animal data that the missing link is this complex AM4 CB1 endocannabinoid cascade, but is that theory the real answer? Or is it just another COX-3, another elegant, promising, and species-specific red herring? We still don't know. The mechanism is still not fully understood. This brings us back to the original question. Why does acetaminophen's messy, 103-year-long identity crisis matter? It matters because it perfectly dismantles the central myth that health influencers use to sell you things. The myth is that medicine is a clean, linear process. Basic science first, clinical use second. The Tylenol story completely dismantles that myth. Its path to our medicine cabinets wasn't driven by a basic science query. It was driven by clinical accidents, expert errors, toxicology studies, pathology reports of kidney failure, and clever marketing. The basic science followed the clinical use by 70 years, and it's still running to catch up. This is the influencer's blind spot. Their entire brand is built on cherry-picking preclinical or basic science data like that 2002 dog study on COX-3 and presenting it as a simple predictive fact. An influencer in 2003 would have told you with absolute certainty Tylenol works on COX-3, and they would have been 100% wrong. They would have found some supplement that inhibits COX-3 and told you that a major breakthrough in pain relief was right around the corner. An influencer today will tell you with that same certainty Tylenol works by activating your CB1 endocannabinoid receptors, and they are making the exact same unproven leap of faith. They are ignoring the long, painful history of failed interspecies extrapolation. More, they do harm by making confident recommendations based on this flawed concept. And how could basic science predict the beautiful complexity of human biology? Just think about how insane the Tylenol story is. Even if we assume that the current hypothesis is correct, no one would have ever invented this drug from scratch. It works by going into the brain, and because it only works there on POX, because there aren't peroxides around, it is broken down into p-aminophenol, a chemical that's toxic in the blood, but just happens to not be toxic in the brain, where it's then changed into AM404. And that substance then acts on multiple different enzymes and receptors. It's a reuptake inhibitor at CB1, and it's an agonist at other receptors, and then somehow all of that causes the release of extra serotonin in the spinal cord in exactly such a way that it blocks pain signals. And this absurd biochemistry is, I'll remind you, the safest pain medication ever created. It's safe for pregnant women, children, and the elderly. It works on everything from post-operative pain to migraine. It has an extremely predictable dose-dependent toxicity, not random like NSAIDs. At normal therapeutic doses, less than 2,000 milligrams a day, it doesn't cause GI bleeding or hemorrhage. It doesn't increase the risks of heart attacks like ibuprofen, and it doesn't even reduce inflammation, which your body often needs for healing. If you were working backwards from receptors, you would have to be a lunatic to propose this mechanism as a good idea. And this is just our best current hypothesis, so we don't have to tell our patients it works by magic. We don't even know if this is the whole truth or if it's true at all in humans. This is one example of how real medicine, real biology, is so fascinatingly complex, and it's why real-world, randomized controlled trials in humans are the only way to know what a supplement, drug, or substance will actually do. There is no way of getting that information sooner. The entire biohacking, personalized medicine concept is flawed, whether it's cholesterol, NAD+, or omega-3s. Extrapolating from theoretical mechanisms and animal experiments is a very low percentage guess of what the actual human randomized controlled trials may ultimately show. We know this looking backward, but we fool ourselves looking forward. The Tylenol paradox is that one of the most common, safe, and effective drugs in clinical history is still an incomplete mystery in pharmacological history. And that is the best reason to be skeptical of anyone who claims to have hacked human biology by reading a study about mouse receptors.